All right, so yeah, thanks to Saber and Eric for organizing this and for inviting me. This is definitely going to be a little bit light on some of the latent space stuff, but I think hopefully you'll see some potential applications for a lot of the things that people have been talking about here. There is one PC state space plot, so you can look out for that. I'm going to talk about speech encoding, which I think is a good sort of maybe starting point for a lot of the topics that people are going to talk about today, because speech is inherently, one of the most complex signals and behaviors that we have. And specifically, it's hierarchical and temporally dependent. And what I mean by that is that, when we speak, we generate these sound waveforms, which we can describe in many different sort of overlapping and complementary ways. So, for example, we can label all of the different phonemes or individual speech sounds. We can also describe these sounds based on how they're produced by the vocal tract. So the colors here are what we call acoustic phonetic features, and this is an example of that. So in the words pin, fin, and fun, they all sort of differ in just a single phoneme, and those sounds are produced differently. So the P sound, this is an acoustic spectrogram, frequency over time. Its characteristic acoustics are these sort of broadband, very temporally modulated sounds, and this is what we call plosives, which is just how the vocal tract makes this. And that would be in contrast to sounds like F, which are sort of these higher frequency, still broadband, but higher frequency and more sort of temporally sustained signals. And we can also contrast other sounds like vowels based off of these sort of characteristic harmonics, which we call formants. And they reflect the shape of the vocal tract. So the auditory system is very tuned to these kinds of sounds, and I'll show some examples of that. But of course, this is not really what we perceive. This is not our experience of speech. We don't hear a sort of string of sounds. What we hear is syllables, words, and phrases. And this is the sort of temporal dependence. And one of the major challenges in speech is trying to understand this temporal integration of how we go from this completely, highly variable, continuous acoustic signal to, potentially these sort of more categorical representations to our ultimate perception, and how this is all happening extremely rapidly. So the main brain area that we're going to focus on is the superior temporal gyrus, which is a core area for speech perception. And one of the reasons that it's important is that it contains these neural populations that are actually very finely tuned to these acoustic phonetic features. So there are populations that are tuned to things like plosives, fricatives, different types of vowels, nasals like M and N. Interestingly, there's not really a map of these sounds across the gyrus, across the surface of the gyrus. They're kind of interspersed. There's not like a plosive region, but this is all embedded in a more sort of broader spatial organization for another level of the speech hierarchy, which is these prosodic features. So things like the posterior STG bilaterally is very sensitive to things like onsets from silence, large changes basically. And the mid to anterior STG is more sensitive to things like envelope, or the intensity of the sound and the pitch of the voice. And this is all going to sort of come back a few different ways throughout the talk. So I just wanted to kind of give this general overview. This is in some ways kind of the state of the art of what we know about speech, primarily from, this is mostly from ECOG data in the last several years. But the question that I'm really interested in here is, how do neuropopulations in STG give rise to our perceptual experience of speech? And what are some of the examples that we can start to understand how we go beyond just sort of these individual speech sounds that are kind of, described out of context? So I'm going to talk about a couple different studies that we've done.
Sorry, it's just, I don't think the microphone works.
Oh.
It's kind of hard to hear.
Sorry, I'm not even talking into it. Is that better?
Thanks.
Sorry. Thank you. So I'm going to talk about a couple different studies that we've done over the last several years that start to get at this question in a couple different ways. And the first one is a phenomenon called perceptual restoration. So this is something that happens all the time naturally when we're just out in the world listening in noisy environments and things like that. And basically the idea is that you can start with a sound like this. This is a spectrogram of the word faster. Again, frequency over time, and hopefully, oh, you know what? I didn't plug this in, but I will. So hopefully you'll be able to hear this.
Faster.
Okay. And what I have here, so what I have labeled is the critical phoneme here, which is the S sound. And the reason that we call it the critical phoneme in the study is because we can take that sound and we can sort of splice it out and replace it with a different sound to produce a different word in English, which is, in this case, factor. So
Factor.
So hopefully you heard both of those as faster and factor. And this is what we call a minimal pair because they differ just in one sound. And what this does is it creates an opportunity for us to then actually take this critical phoneme and just completely remove it from the signal, excuse me, and replace it with a totally uninformative and ambiguous sound, like in this case, pink noise. And so hopefully you'll hear this.
Factor.
So how many people heard it as faster? And how many, did anybody hear it as factor? Okay, a little bit. So this is basically what we took advantage of. And in fact, when you play this over and over again to people, you get behavior like this, which is that 100% of the time when they hear these two original sounds, they'll perceive it as what the actual word was. But on different trials, different repetitions of the stimulus, they'll sort of switch back and forth between hearing it as an S and a K. And so this gives us an opportunity to basically kind of control for the bottom-up acoustic signal because it's identical across these different conditions and look at what the neural activity that's associated with what they're actually perceiving. So we did this study with ECOG, and I'll show you just an example of one electrode from the mid to posterior STG. And this is looking at the high gamma response to the two original sounds, the actual faster and actual factor. And you can see that it's an identical response up until the critical phoneme when you get this difference between the two sounds. And this is actually what we would call an example of like a fricative electrode because it likes sounds like S and F and TH and things like that. So then the question, though, is what happens when we take the trials where they perceive the noise as faster and perceived it as factor and split those? And what we find is that it patterns after the original sounds, and it does it with some interesting dynamics that we'll get into in the next couple slides. But the idea is that when they're perceiving it as an S, it looks more like an S, and when they're perceiving it as a K, it looks more like a K. Even though, again, they heard this is the identical sound across these different trials. So this is just one electrode, though, and what we really want to understand is, first of all, what is the neural representation that's actually changing in these two cases, and also how are the dynamics driving this? So we took a couple different approaches. One of them was looking at stimulus reconstruction. So the idea here is that we have the acoustic spectrogram of these two original words, faster and factor, again, they differ. They're acoustically identical except for the critical phoneme, where you have this high frequency component here for the S, which is not there for the K sound. And the idea is we just use a very simple, multiple linear regression to basically predict this matrix from the neural activity, from the population of ECOG electrodes. And when we do that with these two original sounds, we find that's the difference that we reconstruct. That's the primary thing that's different between them, is that we get this greater power in the high frequency acoustic band for faster, which is not there for factor. And then the critical question is if we do this again, where we split the trials when they heard the noise as one versus the other, what does it look like? And again, we reconstruct that difference as well. So what's interesting about this is that not only is this showing that the sort of low-level acoustic representation is one of the things that's changing across different perceptions of the same noise sound, but it's also happening extremely rapidly. It's not like the S sound is being reconstructed way out here. There's something that's actually happening kind of in real time, and that fits with our experience of this phenomenon. Where out in the real world, when this happens, we're not even aware of it, and most of the time, it basically passes without us knowing. But it does raise the question of what's actually allowing it to happen as quickly as it is. And so we looked at a couple different ways of the population dynamics here. So one of the things that we did was just simply taking the PC state space basically of the different conditions. And first of all, it has this sort of characteristic dynamics that's driven just by the evoked response. So this is primarily STG electrodes, although there's other regions that are involved in this as well. And we find that there is sort of this difference after the critical phoneme, which is shown in the triangles here, But what's also really interesting and stood out to us when we looked at this was the fact that before the onset of the critical phoneme, we're already seeing a difference between the two dashed lines, which are the noise stimulus and the two conditions. And so there's something in the dynamics that's already showing differences in the perception before even the onset of like the critical sound that they're hearing. So what we did then was to try to classify on a trial-by-trial basis what they actually reported hearing each time. And so this is classification accuracy, chance is 50%. This is averaged over many different stimuli. Fast turn factor is only one example, but there's lots of different stimuli. There's lots of different participants here as well. But on average, unsurprisingly, immediately following the critical phoneme, we can classify what they actually heard because the acoustics are different. When we do this for the noise, we also get that effect out here. But again, we see that there's this difference here before the onset of the noise that basically is starting to reflect something that is actually, we would argue is essentially causal in terms of their what they ultimately perceive. And sometimes this is averaged again over many different conditions, and on some individual cases we can classify like 95% accuracy the trials. So this is all just kind of one somewhat isolated example of the relationship between speech processing in the brain and perception. One of the questions, though, that comes about from this is really like, how does this happen so rapidly? In natural speech, things unfold even faster than they do in these sort of single word studies. And so it raises some questions that I think I'm going to spend the rest of the time talking about that I think are kind of interesting and relevant to a lot of the work that we're doing now. So this is kind of like a standard conceptual model of speech processing in the brain that I would say the vast majority of people in the field subscribe to this. The details of it aren't super important, but the point is that at this sort of macro and even the mesoscale with like ECOG, for example, the basic idea is that different brain regions do different things, different linguistic computations. So you'll have a region that's for lexical, like word stuff, and you'll have another region like the STG that is, they call it here spectrotemporal analysis, acoustic kind of things. And this is undoubtedly true at some level. I mean, if you have a brain lesion to a certain region, you can have selective deficits. But I think that phenomena like phoneme restoration and many others that we and other groups have studied, and also just sort of the general rapid nature of speech processing, does raise the question of is the distributed nature of this network really sort of the fundamental feature, or is there more of a role of local computation? And so that's what we're going to get at. And what I mean by that is, if we go back to this kind of overview, we know that underneath each of these sort of symbols here, when we record with ECOG, that of course there's tens of thousands of neurons, and crucially that they're organized into this architecture, which is the laminar structure of the cortex. And so the question that we're interested in is, to what extent does speech encoding in STG sort of rely on or utilize this architecture for local representation of maybe multiple different types of features? Or is it really the case that each of these electrodes can sort of be totally summarized by these different types here, and that they're truly sort of distributed in nature? So to get at this, we've been using neuropixels, which I think everybody here is familiar with, so I'm not going to go into detail on this, but this has obviously been very revolutionary for recording in animals over the last 5 to 7 years. And part of the reason is that it allows for simultaneous recording of lots of neurons, which is great. For our purposes, it also gives us an opportunity to, because of the structure of the silicon probe and the high density nature of it, to record from across the cortical layers if we can insert this into STG. And so this is what we have been doing. We and also others, a group at Sid Cash's group at MGH is working on this as well. And I think we've had some really nice back and forth with them in terms of developing this. And I do want to say that like this for both groups, and I think other groups that are also working on this, this has really been a long process to get this going. I think this is out of date now. We're probably closer to 4 years to be able to really do this safely and effectively. And there have been a lot of challenges along the way, and we've solved some problems, which both of these papers talk about some of those issues and how to solve them. But now we're also running into new things. So that's just to say that this has definitely not been as plug and play as we had hoped. I'm happy to talk to people offline about some of those challenges, and of course, Eric also is the person to talk to about this kind of stuff. So. So now that we're actually able to do this, what are we able to find? And I do want to say that this is all work that Laura Williams, who's a postdoc in the lab, has been working with me on this. She's about to start her lab at NYU in the fall. So what we did was we insert the probe in the OR. This is also, I should say, in tissue that's already targeted for resection. So that's one of the limits that we currently have, is that basically we're only putting this in something that is about 15 minutes after we do these recordings. It's going to be completely taken out. So the idea is that we play these sort of natural speech sentences, like "it was nobody's fault." This is the phoneme, phonetic transcription, the acoustic waveform, the acoustic spectrogram. And then what I'll just show you for an example is one neuron, and they heard this sentence 10 times. So I'll show you the spike raster, and you'll hopefully hear the spikes as well.
It was nobody's fault. It was nobody's fault. It was nobody's fault. It was nobody's fault.
So obviously we're getting really nice spikes from some of these neurons, and they have this very specific firing. It's very characteristic, and very repeatable across trials. So to start to understand what these neurons are actually doing, let's just look at one more example sentence. This is same neuron but different sentence.
Junior, what on earth's the matter with you? Junior, what on earth's the matter with you? Junior, what on earth's the matter with you?
So here we're starting to be able to see that maybe this is a neuron that likes these sounds like N's and M's, for example. This is what we call like a nasal unit. What we want to do is quantify this, of course, because this is just one example, and also I don't want to give the impression that the brain only cares about N's and M's. So what I'll show first is just a whole bunch of neurons, and the details here are not going to be super important because we'll summarize this in the next slide. But what this is showing is 4 different sentences across the columns, and then about 10 or 11 single neurons from a variety of penetrations that we did. Some of them are from the same sites, but the point that I want you to take away is, first of all, again, these are very, very specific in what they're responding to. They're highly repeatable. Each of the rows of the subplots is a single trial. And then the other thing that's really striking about this is the fact that if you look across these neurons, they're essentially tiling all of the different speech sounds. So there's always a neuron that's responding, and this is only just a small number of neurons here, but we're essentially covering the sort of the overall content of the sentences. And so to quantify this, we're fitting these sort of very simple linear encoding models where this is basically the kitchen sink approach. We just sort of take all the different features that we can think of based on previous studies and the linguistic literature, and we annotate the sentences for things like acoustic phonetic features. These are kind of these intensity features like sentence onset and amplitude envelope, and some change in the amplitude envelope, the derivatives and things like that. We also look at the pitch of the voice, as well as some higher order features like stress, so whether a word is actually stressed, and that's of course related to some of these acoustic features, but it's a little bit more abstract. And then we also do include some more sequence level, transition probabilities, biphone, triphone, trigram, whatever, to see if we get any higher order encoding. So we fit the encoding model for each neuron, and then the basic approach is that we look at the unique variants, so we take the full model and we subtract out, or we remove selectively some of these different groups of features, and we look at the contributions that they make. And so this is one site on STG, sort of the more posterior part of STG. And what I'm showing here is a distribution of the unique R² for each of the different types of features. And a couple things stand out. So one is that, and I'll show you actually how much variance this accounts for in a second, but the first thing is that there is sort of this dominant feature in this site, which is pitch. It's not a majority, I would say a plurality, but it is sort of like the strongest feature. The next strongest feature is these sort of acoustic phonetic things, and it's mostly vowels. But what's also interesting is the fact that there is representation of all of these other features. And when we look at the single neurons that are underneath, that go into this pie plot, we see sort of where that's coming from. So there are a bunch of neurons that are, primarily pitch. There's some neurons that are primarily acoustic phonetic. There's a couple that are in the light blue, onset, for example. But actually, a lot of these neurons are also, very heterogeneous in what they're tuned to. So they have multiple features, kind of across this linguistic hierarchy that they're tuned to. When we look at where these neurons are in terms of the depth relative to the surface, we also see that there's an organization here. So, again, these in general they tend to be pretty heterogeneous. But if you just sort of look at the color, what we find is that these sort of deeper neurons tend to be more purple, or have more purple at least, which is these acoustic phonetic features like vowels. And the more superficial ones here tend to be more orange, encoding pitch. And this is actually something that we see in basically all of the sites that have pitch as the dominant feature. This is the organization that we see. So there's potentially something here related to more, maybe these might be, it's very hard for us to know for a variety of reasons, but these might be sort of more inputs that are driving the acoustic phonetic representations, and then these more sort of longer time scale features like pitch, which unfold over the sentence, tend to be more superficial, maybe like layer 2, 3. Just to show you what this looks like across these different sites, this is that sort of large pie plot for each of the sites that we recorded from. And again, what you can see is that each site kind of has a dominant feature. So these 4 here are primarily pitch. These 4 would be primarily some acoustic phonetic stuff because of the purple, and then there's one here that's primarily onset. But they are still, so basically they have this dominant feature, but they also are, kind of surprisingly heterogeneous. And I'll explain in a second what I mean by surprising. But I also just want to emphasize that, this is true both at the population level and at the single neuron level. So the next thing we did want to look at was the laminar organization, and to see if we could understand a little bit more about what's going on within this sort of circuit of a putative column. And the idea here is that earlier areas in sensory cortex, including primary auditory cortex, are characterized by things like ultra-narrow frequency tuning. This was a study from 15-ish years ago from UCLA, where they showed that primary auditory cortex neurons are very narrowly tuned. And when you look at these kinds of neurons across the cortical depth, this is in cats, you see that they're basically, this is sort of like the Mount Castle cortical column idea here, in the sense that they're all tuned to essentially the same thing. And we wanted to ask, to what extent is this the same in a higher order area like STG? So we fit these models, instead of using the sort of linguistic descriptions, this is using spectrotemporal features, so it's just the acoustics basically. And there's a couple things that stand out. This is 6 neurons from one site across the depth. And basically, first of all, these are very broadly tuned. These are not primary auditory cortex neurons. They're tuned to things that are really sort of remarkably complex in terms of the acoustics. Some of them have this sort of broadband tuning. Some of them are more sort of like spectral modulation kinds of things. So that's one difference that we're seeing. Another difference is that when we look at, this column is another set of 6 neurons which are just very adjacent to those same neurons at different depths. And we see that, first of all, they're very similar to the ones that they're close to and different from the ones that they are further away from in terms of the depth from the surface. And so we quantified this by basically just taking the correlation between these matrices in this sort of pairwise fashion. We binned things into different depth bins to simplify this, and this is what we get. So the correlation between the STRF weights and the fact that there's this sort of strong diagonal tells you that neurons closer to each other have more similar tuning, and neurons further away are less similar. So there is already the sort of organization that we're seeing across the depth, across these cortical layers. And then the last thing that we wanted to look at here was basically, do we see any evidence that this reflects some sort of microcircuit type thing? And so basically we just started by looking at the latencies of these representations, basically the peak lag of the STIRF, which is on the x-axis here. The depth is on the y-axis. You can see that there's a lot of variability in terms of when these neurons are encoding this information. But one thing that does stand out is that the only area that's sort of consistently less than 100 milliseconds is this sort of mid to deep region, which would be consistent with kind of thalamic inputs. So the last thing I want to talk about just in the last minute here is what is the relationship between all of this stuff that we're seeing and what we've been doing for the last 10, 15 years, and others have been doing a lot longer with ECOG. So we have this sort of unique opportunity to directly compare these in a couple of cases where the patients will be in the epilepsy unit, inpatient for 7 to 10 days or so. We can record with ECOG electrodes on the surface. If we identify a site that's interesting and is also then targeted for resection, they go to the OR and we can put the NeuroPixels probe in for 10, 15 minutes and record. And this allows us to basically ask what is this relationship between neurons at different depths and what we've been seeing with ECOG. And there's a couple different, kind of obvious hypotheses here. One is that maybe it's the most superficial cells, the ones that are just closest to the electrodes and to the surface contacts. Another is that it might be the thalamic inputs, or the cells that are the most strongly phase locked, or also maybe the cell bodies that are giving you the strongest signal. And another possibility is it's kind of everything, that we're seeing contributions from neurons across the whole depth. So this is, these are just PSTHs of 2 example sentences. In the red is the ECOG electrode that's directly over the NeuroPixels recording site. And in the purple is just the straight up average across all the neurons at that site. And what we see is that they're extremely highly correlated. So there is definitely a relationship here. This is the high gamma signal of the ECOG, and I'll talk about that in a second. But there's definitely a relationship here. And so the question then is the relationship similar for neurons across the depth? And what we see is the correlation between ECOG and each single unit is on the x-axis. And we do see that basically the answer is yes, that neurons across the entire depth are quite strongly correlated with the ECOG signal, specifically the high gamma signal. If we compare this to the LFP of the ECOG, we see that it's much stronger for high gamma. And if anything, actually, so it's the whole depth, but if anything, actually, there's potentially a greater contribution of these sort of mid to deep layers that we were seeing. So it's probably some combination of this sort of everything hypothesis with maybe the thalamic inputs. So just to summarize the last slide here, a couple implications of this. One is that so the rapid nature of speech perception, I think we're starting to see. It really does take advantage of these sort of local computations and this structure that's available within single neuronal populations in cortical columns, to the extent that we want to call these columns, which we can of course talk about. These columns, they do reflect what we see at the ECOG level. This dominant tuning, the sort of overall population response is certainly true, but we're also seeing now that they contain neurons that represent many different types of speech information, and it's quite heterogeneous. This probably goes without saying to this crowd, but for people who work in this area, I think this is somewhat of a novel idea, is this idea that cortical layers really are a crucial computational dimension, and we need to start to understand them. And this is kind of one of the only ways that we have to do this in humans at the moment. And then just sort of looking ahead, the idea that this ability of what we're able to do with these NeuroPixels probes in humans is really opening up a lot of new possibilities for understanding these circuit level computations, which hopefully is going to get us a little bit closer from what's been the dominant way of studying this in humans with fMRI and EEG and things like that, to something that's closer to what can be done in animal models and at that level of resolution. So with that, I just want to say thanks to all of the people who've worked on these projects and to our funding sources. So thank you.